Hello, everyone. Hello, everyone. Today, we will be talking on antigens, that is the biological classes of antigens. Those who want to know more about antigen can visit my previous videos, whose description I will be putting in description boxes. But today, we will be continuing on biological classes of antigen. This is the way the antigen, when enters inside the host, how it behaves and what responses it gets from host. One is T cell dependent antigens. Here the T stand for thymus. So thymus, that is T cell dependent antigens, one are those and other are T cell independent antigens. So knowing more about T cell dependent antigens, as the name signifies, here the help of T cells are required. T helper cells are required. B cells alone will not be able to respond. Since both the cells are required, so this kind of antigens contains two types of epitopes. That is, some epitopes which will be recognized by T cells and others which will be identified by B cells. When T cells also recognize and then T cells give signal to B cells. This is a very important and interesting way how antigen is presented and what happens and how T cells help in production of B cells. Here we are talking about antigens, so we will learn only the type of antigens which will provoke this kind of response. But how this response takes place is interesting, and as I have promised to you all, I will make it more simplified as possible. And so we will be reading in next lectures on immunology. And that will be very interesting how antigen is presented. Here, since we are concentrating on types of antigens, so here we will just talk about these antigens, those who will be recognized by T cell receptors as well as B cells. And what are those antigens? They are most of them are naturally occurring proteins. So naturally occurring proteins belong to the class of antigens where T cell dependent these will be called T cell dependent antigens, and here the need of help of T cells will be needed. What kind of immunoglobulins or antibodies these T cell dependent antigens can trigger? This is they can induce IgG, they can induce IgM, they can induce IgA, they can induce IgD and IgE synthesis. That means all 5 types of immunoglobulins can be induced by this T cell dependent antigens. Next class is T cell independent antigens. As the name signifies, there no help of T cells will be required. B cells will be enough to identify these antigens and respond to it. So what kind of antigens are there? They have the repetitive epitopes. Since they have repetitive epitopes, that means they may be similar kind of molecules joined together, and they usually induce synthesis of IgM. Basically, not all 5 types. There are 5 types of immunoglobulins. If you want to know more about immunoglobulins, you can visit my site where I've uploaded videos on immunoglobulins too. So here, just to keep myself in check, is that IgM will be synthesized by B cells in the class that is T cell independent antigens. And these T cell independent antigens do not stimulate immunological memory also. While in case of T cell dependent, this point is not included in the slide. But remember, when T cell dependent antigens are there, they will act as memory cells. They will be carried as memory. So T cell dependent stimulate immunological memory, while T cell independent do not stimulate immunological memory. Most microbial sugars like lipid and nucleic acid belong to this class. So these are the examples of T cell independent. T cell independent antigens are of 2 types, type 1 antigen and type 2 antigen. So T cell independent can be classified into 2 types. That is, type 1 is the one which induced polyclonal antibody production. That means B cells of different lineages can produce antibodies in this case. So that lineage, when B cells of different lineages can produce antibodies, and it's just mitogenic in nature. They will just multiply and produce antibodies. These kind of antibodies are produced by type 1 antigen, that is T cell independent type 1 antigen. And most of them, the example of this type 1 T cell independent antigen is lipopolysaccharide, that is bacterial cell walls, usually bacterial components of bacterial cell wall. Talking about T cell independent type 2 antigen is the one which is prolonged and persistent, which gives prolonged and persistent signaling to B cell. So this signal will be for a long time. And stimulation of non-T cell, that means their T helper cells are not required, but this type 2 antigen is the one which will activate complement and other dendritic cells, mast cells, and natural killer cells. So help of these cells will be needed. The type 2 is the one which is prolonged and which is causing prolonged and persistent signaling, and because of the prolonged and the persisting signaling, B cell also signal to some other non-T cells and interact. These cells then will start interacting non-T cells. Here T helper cells are not needed since it is T cell independent. So what happens? Other cells will be needed in this case, and these other cells will interact with B cells, because B cells are the one which are going to synthesize antibodies. Other cells will interact with B cells, and this cell talk is through whenever there is cell to cell talk, it is through cytokines talk. So a lot of cytokines production will take place in this case. And what are the examples of T cell independent type 2 antigen? It's usually repeating polymers like dextran, ficon. Today we will be talking only on this. Then in the next lecture we will be discussing super antigens. Thank you so much.